right, so here I'm going to talk a little bit about restriction enzymes. Okay, restriction enzymes are bacterial enzymes. They are endonucleases. The function and purpose of these restriction enzymes is that they are able to recognize and cleave specific sequences. These restriction enzymes are widely used for cloning because these enzymes can be used to prepare DNA fragments containing specific genes or portions of genes. So when a restriction enzyme does its job, because DNA is anti-parallel, whatever sequence is cut out will also be present on the complementary strand of DNA. There are two different types of cuts that these restriction enzymes can bring about. And here in particular in the picture, we see sticky ends. Okay, and these sticky ends come about because the restriction enzyme hydrolyzes the phosphodiester bond between the nitrogenous base residues. So sticky ends are brought about because there is a hydrolysis of phosphodiester bonds between nitrogenous bases. Okay, the second type of cleavage associated with restriction enzymes is what are known as blunt ends, okay? And these blunt ends differ from the sticky ends in the fact that the reaction occurs in the common structural component of DNA, which is the phosphate sugar backbone, instead of the actual bases. So therefore, that the information in the DNA, that is the sequence of the nitrogenous bases, is preserved. Okay, so how did these restriction enzymes come into play in the whole microbiology, biotechnology field? Okay, there was a couple of scientists. I'm not going to say their names. Yes, they're important. But basically what happened is they were doing an experiment and they went and announced that they had successfully generated recombinant DNA molecules. And how they did this was because they allowed the sticky ends of the fragments that a restriction enzyme had cleaved. And basically what happened is that these sticky ends were allowed to anneal. And that is each end was allowed to base pair with the other end, and these were covalently joined by the enzyme DNA ligase. Okay, this paved the way for inserting these genes into cloning vectors, and biologists were trying to look for ways to clone specific genes from various organisms. However, they came into a little dead end when they saw that cloning eukaryotic DNA into bacterial hosts was a little problematic, because eukaryotic mRNA must undergo a process such as, the introns are spliced out. Bacteria lack that molecular machinery. They are not able to perform that task. So, they went and they did some discoveries and they found the enzyme that solved that dilemma. The enzyme that was going to allow bacteria to perform that function of splicing out the introns in the eukaryotic DNA. So basically, how they solved this dilemma was by isolating an enzyme. And that enzyme was the reverse transcriptase, which was isolated from retroviruses. So basically is these viruses have an RNA genome that is copied into DNA. That's why it's reversed, because usually it's DNA to RNA, but in the reverse, you're going to get RNA to DNA. So then it's not an actual DNA molecule per se. It is basically a processed mRNA that is used as a template for a complementary DNA, which is a cDNA. So the cDNA is basically able to undergo cloning, and it doesn't need to go through all the RNA processing steps. But reverse transcriptase is totally another topic, and we'll talk about that in more detail in another video.